Okay. All right, welcome everyone to the muscle lecture. If you will begin in chapter 14 on page 535, the muscular system. We'll start with the characteristics of muscles, how muscles, then we'll move on to how muscles work, what the theory behind muscle building is, and the aging process of muscles. Let's start with the characteristics of muscles, right? Muscles make up about 50% of our body mass. So it's quite a bit of us. If you think about our skeleton, the skeleton is tiny. And often when you see that just a bone, go like so, and just feel your bone, there's not much to it from here to here. And when you think about it, we're little creatures. If the flesh were not around us, which is the muscle, the fat, and all of that connective tissue. So muscle makes up quite a bit of us. Muscle is unique in that it's able to transform ATP energy into mechanical energy. And what do I mean by that? ATP, or stored energy in the phosphate bonds of ATP, get turned into mechanical energy. And if you think about it, mechanical energy is, mechanical movement. When we did the dollar bill drop in an earlier lecture, I kind of wanted to show you how your brain sends electrical impulses to your hand to move it. The movement of the hand is the mechanical energy. It takes ATP to do that movement. And what our agenda is in the next lecture is the nervous system. How do you move the muscle? When we go through the muscle, always have in the back of your mind that every muscle is moved by a nerve stimulating it, just like an electrical wire stimulating all the muscles. Okay, we've got three types of muscles: skeletal, cardiac, and smooth. We have talked about that in the histology lecture. You know that the cardiac muscle is the heart muscle, the smooth muscle is on the organs, and the skeletal muscle is on our bones. Our focus of today's lecture is primarily on the skeletal muscle, right, and what its purposes are and its general structure. So again, when we think of muscles, we think of movement. You catch the dollar bill, you move your arm, you do whatever it is that the body wants you to do to have mobility. However, we often do not think about the posture muscles. We call them postural muscles. Muscles of posture are slow to fatigue. They are also known as, and we'll discuss this later in the lecture, slow twitch muscles. They're slow to fatigue. Posture muscles are muscles of your back, your neck, muscles in your leg to keep you standing, to keep you sitting, and not collapsing and falling over. These muscles are structured and have a different metabolism. They're structured a different way, have a different metabolism than faster twitch muscles, or those that are more fast to fatigue. They keep you standing, they keep you sitting, otherwise you would plop over. And then you have to always appreciate your postural muscles. You don't think about it. If you didn't have these postural muscles, you would flop over. I guess the ideas behind yoga and Tai Chi and these types of training is to strengthen your postural muscles. In other words, strengthen your core. Your core muscles are those muscles holding up, the axis of your body. As you know, in the labs, we talk about the axial skeleton. That's, of course, primarily your backbone holding you up. So the muscles around that and the ligaments and tendons around that are your core. We also say that muscles stabilize joints. In our orthopedic unit, which this is, we emphasize that the joints, your elbow, your knee, your shoulder, you can't do anything, as we said, about your cartilage. You're born with a certain type of cartilage, your ligaments, your tendons, you have a certain structure. What you can definitely strengthen is the muscles around it. And the whole point of physical medicine is to strengthen the muscles around those joints so that those joints are less prone to damage and are more mobile. Last but not least, sphincters and the generation of heat. Sphincters are round muscles. Look at my finger, round muscles. They open and close. You have a sphincter on the top and bottom of your stomach to keep the acid in and let let food in and let food out. Sphincters are guardians of entrances and exits in the body. I gave you one example, the stomach. There are other examples, but that's a sphincter, pronounced SPH with an F, sphincter. Generation of heat. Muscles generate heat. Muscles have a lot of mitochondria, and mitochondria produce heat to maintain your body temperature. Muscles have a lot of mitochondria because what do muscles do? Turn ATP energy into mechanical energy. You need a lot of ATP in your muscles to perform the functions of those muscles. We say the characteristics of muscles are the 3 E's and a C. As you can see, excitability, extensibility, elasticity, those are the 3 E's, and contractility is the C. Why don't we go over the 3 E's first? Excitability is just what I mentioned a few minutes ago. You excite a muscle by stimulating it with an electric wire. That electric wire in your body is the nerve, a neuron. Topic of the next class. I remember getting, my father's an electrician, retired, you know, and I was his helper many years down in Corona. You know where Corona is, anyone? Let's see if someone knows. Corona, Queens. He was Bell Electric, and I was his helper, you know, just a little bit. I mean, I was a science nerd going to high school, college, and I just want to say I was electrocuted 3 times in my life, and that explains my personality. I'm just kidding, but anyway, I was electrocuted 3 times, and they weren't bad, but you can feel it. It is a numbing. I once was putting in a baseboard heater, and you know, touched it. My father left it live, you know, because he does it live, and I am not an expert. And it went right down my ulnar nerve. Whoops, I'll show you where that is. Ulnar nerve right along here. As you know, the ulna, you've been studying that, right? That's this week's lab. This where your ulna is. Well, the nerve goes along it, and it just numbs it, feels hideous, and just paralyzes it. I hate it. So anyway, that's excitability. Excitability leads to the next thing, contractility. You basically shorten it. A muscle shortens in order to move things. I repeat, you should write this down, put a little star next to this. A muscle shortens when it is excited, and that's what moves things. So muscle shortens when excited, and that's what moves things. It is extended when it relaxes, so it stretches when it's relaxed. And then it's elastic. Elasticity is the ability to resume length after a contraction. So that's elasticity. It can go back and forth like an accordion. I always think of it as an accordion, like so. And the little filaments slide like accordions in the muscles. And what is an accordion? You know, a musical instrument where, you know, you move it back and forth. You can stretch it and push it back. My brother used to play an accordion. Anyway, I was not musically inclined. Here is the structure of a muscle. Here, let me get myself out of the way. A muscle and its connective tissues around it. This is the gross anatomy of muscle. How exciting, right? I mean, seriously, this is a microscopic picture so that you know we're not making up a cartoon. As you can see from the microscopic picture, there are little muscle fibers all around. Let's take a look, and let's take a look at the cartoon better. This is a muscle, all right? This happens to be the brachioradialis muscle, and you'll learn that this week, right? When you go over the muscle lab. That's the brachioradialis. Now we're blowing that up, and you're looking at the whole muscle. The muscle is made up of wires, and more wires. And what you have to know, you got to know this picture. You have to know the general structure of the muscle. Every wire has to have a covering around it. Otherwise, you can get yourself electrocuted, right? You need a protection. And same thing with the muscle. You need a protection around it so that it doesn't get damaged, and also to keep the charge inside. That is true after it is electrically stimulated. So the epimysium is the connective tissue that surrounds the entire muscle. Epi means around, right? and that connects to a tendon. A tendon is an indirect connection of the muscle to the bone. Okay, so the epimysium. Got to know the word epimysium. So circle that, got to know it. Then what does it surround? The entire muscle. So here's the whole muscle. What's the muscle made of? It's made of bundles of muscle fibers. All right, bundles of muscle fibers. And these bundles, that's one bundle. Look, 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 that's another bundle. That's another bundle. That's another bundle. Each bundle is known as a fascicle, all right? This is a fascicle on the microscope slide. This is a fascicle, this is a fascicle, this is a fascicle. That fascicle is surrounded by a perimysium, as you can see. See this little picture? These are all coverings of the fascicle. So a perimysium covers the fascicle, right? And the fascicle is made of individual muscle fibers. So if you pull a fascicle out, you see that? That is literally a pulled out fascicle. Again, fascicle is bundle of muscle fibers. You pull that bundle out, and then let's pull out an individual muscle fiber. Can you see that? I think so. Individual muscle fibers. Those individual muscle fibers have a certain makeup. The muscle fiber, the word muscle fiber is equal to muscle cell, okay? I'll write that down in the chat room. Muscle fiber equals muscle cell, all right? I'm gonna post that just so you see it. Muscle fiber equals muscle cell. So what are we gonna do now? We're gonna look at the muscle cell. What does the muscle cell look like, all right? Again, this is my little cartoon. You can draw it yourself. Draw a little picture of this general structure where the epimysium, right? So that's the epimysium, surrounds the whole muscle. The muscle is made up of fascicles. Each fascicle is made up of muscle fibers. Each fascicle is surrounded by a perimysium, and each muscle fiber is surrounded by an endomysium. That's the one thing I didn't mention. The endomysium surrounds the muscle fiber. Why, you're saying? You know, why bother? Well, the epimysium has to have dense irregular connective tissue covering and protecting the whole muscle. The perimysium similarly covering and protecting the individual fascicle. But the endomysium, it doesn't just protect, it really organizes capillaries and nourishes the muscle cell. The endomysium surrounds the muscle cell, and that's what we're going to be talking about. I'll have a little hierarchy to help you remember what's bigger than what. And a muscle is, of course, bigger than a muscle fiber. A muscle fiber is made up of myofibrils, and myofibrils are made up of sarcomeres. A lot of words, but I'm going to show you what the bottom two words are in a moment. I want to stop for a minute and see if you have any questions. I'll open the chat room and again go over these connective layers, all right? Just want to go over these connective layers once again. And let me state, the epimysium surrounds the whole muscle. The perimysium surrounds the fascicle. Both of those are merely protective, fibrous connective tissue. It's the endomysium that nourishes, and when I say nourishes, I mean has capillaries that bring nourishment to the muscle fiber. So the endomysium surrounds the muscle fiber, in other words, the muscle cell. Okay. That's a difficult concept. And I think if you just get yourself, you know, your charger cord and you just visualize it as a muscle with many little layers, that would help. Let's take a look at the muscle cell. The muscle cell itself is made of the last two words that I mentioned. I know you're like, what is he talking about? Myofibrils, sarcomeres. Here's what I'm talking about. When we take the muscle cell and we pull out the fascicle and then pull out the muscle fiber, we take the actual myofibril. So again, let me repeat that. I may have misspoken. When we take the whole muscle, pull out the fascicle, then pull out the muscle fiber, right? Here's the muscle fiber. So again, whole muscle, fascicle, muscle fiber. This is the muscle fiber or muscle cell. This is what it's surrounded by. It's surrounded by an endomysium, and then the cell membrane of the muscle fiber has a fancy name. It's not called a cell membrane. It's called a sarcolemma, everyone. A sarcolemma is the cell membrane of the muscle fiber. And that's what surrounds the muscle cell or muscle fiber. And then it's made up of many little myofibrils. Myofibrils are pure protein. You want to build up muscle, you got to eat more protein. You're supposed to have 10% protein in your diet. You know, you have 100 calories, 10% is supposed to be protein. So you want to build up, you got to put 12 or 13%. All right? Because look at these muscle fibers. They are made up of 90% myofibrils. And myofibrils, let's take this little tube and blow it up here at the bottom. This is what a myofibril looks like. A myofibril is chopped into individual little sarcomeres. Sarcomeres are the functional or contractile unit of a muscle. They contract on their own, each individual one. And this is the structure of a sarcomere. I always have that on the test, so you got to know it. I'm pretty sure it's on the test. I gotta double check, but I'm pretty sure it's on some test. I think it's on the next test. Sarcomere is the area in between the two Z discs. All right, what am I talking about? Again, let me just reiterate. This is your muscle cell, right? We pulled out an individual myofibril. In other words, a strand of protein in the muscle cell. And I want to show you what it looks like. That strand of protein is made up of these two names. The strand itself is called a myofibril. And if you chop, all right, let me show you me. Chop the noodle, this little noodle, then each chop is a sarcomere. In other words, the area in between the two Z lines is the sarcomere. That's called a Z line or a Z disc. Okay. And what is all this made of? It's made up of two proteins, actin and myosin. Actin is thin. You can remember that. Repeat after me. Actin is thin. Actin is thin. The little thin blue protein is actin, and the thick one is myosin. And myosin has little heads. I think I asked you that on a test. Myosin has little heads that want to touch the actin. And that's how the muscle moves. The little myosin head attaches to the thin actin molecule, and it swivels and pulls it together. And I will tell you about that just a moment. The functions of the wrappings: protection, insulation, nourishment, organization. All right. I just want you to realize that is the functions of how and why we have all these wrappings around the muscle. I'm going to discuss the structure of the sarcomere in just a moment. All right. Any questions for me on this thus far? Feel free to send a chat question and I'll get notification. All right. Okay, moving along, muscle-bone connections. I told you most bones indirectly attach with a tendon, right, onto a muscle. Most of the time, you don't have a direct muscle-bone connection because you would rip and tear at that very delicate muscle. Muscle is very expensive to make. As you can see, it's quite complicated, right? Connective tissue wrappings, proteins, all kinds of stuff. So you don't want to mess with it. You want a tendon, right, to attach to the muscle from the bone to have a little more give and not rip and tear on the muscle so much. And an aponeurosis is a band-like tendon. So that's a fancy word. Your frontal aponeurosis is right here. Let me show you. Right here is your frontal aponeurosis. A tendon, you have a ton of tendons around your shoulder. So those are two examples of muscle bone attachments. Let's talk now the arrangement of the fascicles. When you look at a muscle, and I'm sure if you turn to page 540, 541, figure 14.28, you've got all your muscles there. I have a nice picture there of all your muscles. You will see that there are little lines. Those lines are your fascicle arrangement. And there's 4 types of fascicle arrangements in the body. One, let's start by feeling our biceps. Your biceps are parallel, all right? So these are your biceps. They're parallel muscle fiber arrangements. In other words, What do you do when you exercise your biceps? You do a curl. So all of the fibers are going in one direction. All right, all the fibers are going in one direction. And you exercise always along the axis of the fibers. So if you're going to work out your parallel muscle arrangement, you got to work it out in that direction. All right, so that's why a curl is like so. Okay, then so that's parallel fiber arrangement, all go in one direction. Pennate means like a feather. The arrangement of the fibers all come together at one line in the middle. That's a pennate organization, and your rectus femoris, in other words, your muscle that is in your leg, right? Your upper leg has a pennate arrangement like a feather, and it can go either direction. Convergent is when you do a bench press, right? Or a push-up. Convergent. All of your pectoralis major fibers come to a certain point. They all emanate from here, right, up to one point and make your humerus move toward the midline. We say that that's called an abduction, move toward the midline. They converge to a certain point. Now, you get my drift? The arrangement of the fascicles tells you the action of the muscle. When we say action of a muscle, that means what it does. What is its action? What does it do? Circular arrangement of fibers, is your lips, your eyes, close your eyes, open and close your lips. It forms a circle around the body of an opening and opens and closes. That is what a circular muscle does. The orbicularis oculi of the eye is our main example. All right, so I'll give you an example of each, and it makes common sense, right? That the arrangement of the fibers dictates how the muscle moves, always along the way in which the fibers are. So it's a little muscle anatomy. When you go to the gym, when the gyms open up, you can talk muscle anatomy with people. You'll make a lot of friends and probably more enemies, though. People do not like to hear about fascicle arrangements. Not at the gym. Trust me, I know. All right, so moving along, muscle fibers. Let's just, I just want to go over what the muscle fiber looks like, right? This is not your average cell. An average cell is a round little thing, right? A round little thing, 80 micrometers wide, and this is not a muscle. A muscle can be up to 100 micrometers in diameter, 30 to 40 centimeters long. So you have some long muscles. It's the fusion of many embryonic cells. So you have long cylindrical cells, and as I mentioned in histology, there's multiple nuclei in the skeletal muscle. So that's to control such a large cell. And the muscle cell is surrounded by a sarcolemma. Again, a cell membrane around the muscle fiber. Lets things in and out. That's just your cell membrane. And sarcoplasm is the special cytoplasm of the muscle fiber. So you can't say cytoplasm and cell membrane. You have to say sarcoplasm and sarcolemma, all right? And why do we say that? Because it is a different type of cell. It's a special cell. The cytoplasm of the muscle has a lot of glycogen and myoglobin, all right? Glycogen is the storage form of sugar, and glycogen allows you a lot of instant energy in the muscle so the muscle can do what it's supposed to do. The muscle cell needs a lot of instant energy to change the ATP into mechanical movement. Myoglobin's interesting. How many can go for a long time underwater? Think about that. The more muscle you have, generally, the more myoglobin. Like a seal has a lot of myoglobin. It can dive for a long time. Myoglobin is like hemoglobin. It stores oxygen. It's a red pigment that stores oxygen. And the longer you can go over underwater, generally, the more myoglobin you have. So myoglobin is an extra strong holder of oxygen, and it sits in your muscles so that you have instant access to oxygen. And you need that all the time to get energy out of food so that you can contract. That's the main purpose. So again, both of these, you got the food, glycogen, you got the oxygen, all so you can have instant access to food and oxygen to contract. Now let's look at myofibrils. Myofibrils are just the actual protein of the muscle. And there's two, again, I'm going to repeat the two words that you have to know for protein. Actin and myosin are the two proteins that make up the muscle. And 80% of the muscle by volume is actin and myosin protein. And the mitochondria, as you can see, are sandwiched in between. This is a good picture of it. You have a nucleus, another nucleus, and here's the little mitochondria, dot, 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 dot. They're sandwiched in between, a lot of them, in between the main thing, the muscle proteins. Now, these muscle proteins, you've got to remember this structure. This is the sarcomere. What we're doing, we're pulling out a muscle protein, a myofibril. It's another word for the muscle protein. The thick bands are the myosin. The thin bands here are the actin. So I want to say that the area in between these Z discs, between this one and this one, is a sarcomere. So that's one sarcomere, another sarcomere, another sarcomere. Again, this is a cartoon of it. Here's your Z disc. It looks like a Z. Z in German, it means zusammen. Zusammen means together, and that's why it's a Z. But it looks a little like a Z. Look, Z. So if you look at the Z, you can always remember that's your Z disc. The area in between two Z discs is your sarcomere. And your sarcomere is made of two parts, actin and myosin. The area where there's all actin and it looks very thick under a microscope is called the A band. The A band is the thick band, and then this band, right, where there's just actin, that's the I band. A stands for anisotropic, I stands for isotropic. You don't have to know that, but you got to know the A band is the thick area, the I band is the thin area, and then the Z discs are the border between one sarcomere and another. The M line makes common sense. That means M for middle, it's a line down the middle. The H zone is this, between one side of the M line and another. The H zone is the area where there's only myosin, right? Only myosin. So let me repeat this again. The I band looks thin and has just actin. The A band looks thick and has myosin. The M line goes right down the middle. The Z disc is the border, and the H zone goes down the middle and goes on either side of the M line and is just the myosin. Now, our purpose is to talk about the sliding filament theory, how a muscle contracts. And I've got a picture, page 536 of what we just talked about, and a list of things that happen during muscle contraction. Figure 14.25 of the book shows the steps of muscle contraction. So maybe go to 14.25, page 538. The first step in a muscle contracting, just close your eyes, visualize. How do you get your muscle to move, right? Your arm to move. Action potential. The word action potential, you can write down next to this, is nerve, right? Nerve impulse. So a nerve stimulates. The word action potential is actually a fancy word for nerve stimulation. So nerve, maybe you're thinking about your arm moving, you're sending a message to your arm. That's step one, action potential. And I wrote the word ACH here. ACH is acetylcholine. It's a chemical that stimulates the start of the muscle contracting, right? Here's the word, just so you know how to spell it, acetylcholine. That is what is given off by a nerve. It's a neurotransmitter, which means a chemical from the nerve. We're going to go over that in the next class. But it's a neurotransmitter that basically is released from the nerve and hits the muscle. Whoa, that causes an earthquake. Earthquake. And that's step two. The earthquake is the nerve shaking up the muscle. And step two, what happens when a nerve shakes up a muscle? Calcium is released from the endoplasmic reticulum of the muscle. we call the endoplasmic reticulum of a muscle, if you look on figure 14.25, the blue stuff is the endoplasmic reticulum of the muscle. That's what stores the calcium. We call it sarcoplasmic reticulum. So you got to have a fancy name for the muscle. It's a special cell. It has high priority. So the sarcoplasmic reticulum holds the calcium. And when the nerve impulse comes, it shakes it up. Whoa. And calcium leaves the sarcoplasmic reticulum and flows from the sarcoplasmic reticulum into the sarcoplasm. That's step 3. Step 4, the calcium combines with troponin. Troponin is a little button, a chemical on the actin that covers up a little hole. Calcium combines with the troponin, makes that a troponin, something called a troponin-tropomyosin complex, which covers up the myosin, makes it shift. It's a complex on top of the myosin that covers up, troponin tropomyosin is a covering of the actin, and calcium attaches to it, makes it shift, and reveals something called myosin binding sites. Myosin binding sites on the actin are revealed so myosin heads can attach to the actin. And then we get the power stroke. The power stroke means the myosin heads swivel and push the actin away, and that causes a contraction of the muscle. I'm going to show you something very shortly to help you visualize and understand this. And last but not least, last step of the sliding filament theory is relaxation. ATP has to actually come in and relax the heads of the myosin. When you teach something like this is very difficult for a student to understand, because figure 14.25 gives you a cartoon. I gave you the steps. I went over it, but you're like, what's the guy talking about? I know you're thinking that. So I have a little moving cartoon to help you remember. Let me get that out in just a moment after I discuss relaxation. Then I'll show you the whole step. Muscle relaxation is achieved by getting rid of the acetylcholine. So acetylcholine esterase is the chemical that gets rid of the acetylcholine and stops the action potential. You got to stop the action potential, right? So acetylcholine esterase is what? An enzyme, ASE. It breaks down the acetylcholine. So calcium release channels close, no new calcium flows in, no more acetylcholine, and you've got to get rid. So right, this is a little picture of acetylcholine and the nerve stimulating the muscle, M for muscle. There's your little nerve. It gives off acetylcholine. You got to stop the acetylcholine. You got to break it down into acetic acid and choline, and you do it by the enzyme acetylcholine esterase. You also have to get out calcium. Active transport pumps pump out calcium from the muscle, and calsequestrin is a calcium binding protein that acts like a sponge to suck up the calcium. All right. What I want to do now is I want to show you a little visual of all this, and then we'll go from there. Okay. It's better than I can do. Hopefully we Merriam's sliding filament theory of muscle contraction is a very nice video. I have to look for it on YouTube that really helps. And sometimes I assign it for you to just muscles. Let's see. Sorry about that. Let's see. Hope they didn't take it off. I used it not long ago. Sorry about that. I just want to show you this. I thought I had this. Uh-oh, that's really bad. I'm in the middle of a lecture. I can't. It's not good.